Let's take a look at ribosomes. This cell organelle is specifically involved in protein synthesis, and we'll actually say that it's the manufacturing plant of proteins within the cell, and that's because it is the site of protein synthesis, and the process of protein synthesis is of course transcription. And the ribosomes are located within the cytoplasm of the cell. So if we look at this diagram at the bottom, these small red dots are showing the ribosomes within the cell. And the number of ribosomes is usually in the millions, and the specific number depends on the level of protein being produced within the human cell. And that's obviously going to depend on the specific organ that the cell is located in. We've got another diagram here on the right hand side which shows the structure of a ribosome in a bit more detail. So at the top we've got what we'd call the large subunit of the ribosome, and at the bottom we've got the small subunit of the ribosome. And we can see in the middle that there's three other sites that are relevant specifically within transcription. So let's go ahead and label these. On the right hand side we've got what we'll call the A site, which is also known as the amino acyl tRNA site. In the middle we've got the P site, also called the peptidyl tRNA site, and on the left we've got the E site, also known as the exit site. And as we mentioned, all of these sites are very important in the process of transcription, in the process of producing proteins itself. Now back to our subunits. The large and the small subunits actually have a specific unit of measurement, and the large subunit we can say is 60S, and the small subunit we can say is 40S. Now you're probably wondering, what does this measurement mean? What does this S mean specifically? Well, the S is Svedberg units, and this measurement doesn't actually measure the mass or the size of each subunit. It measures the rate of sedimentation in ultracentrifuge. So it measures the rate at which the different parts of the ribosome break up in centrifuge. Okay, so let's look at the structure of the ribosome in a bit more detail. We've got two main components of the ribosome. So we talked about the large subunit and the small subunit, but what are they actually made of? Well, the first of the components of the ribosome is ribosomal RNA, or rRNA for short, and this forms about 60% of the total mass, if you like, of the ribosome. And specifically, the sites of transcription, remember we talked about the A site, the P site, and the E site. These are mostly made of ribosomal RNA. Now the second component of ribosomes is proteins, and specifically about 80 different proteins, and these form about 40% of the structure of a ribosome. Now the proteins, they actually are usually located on the surface, whereas the ribosomal RNA forms what we call the core, so the central part of the ribosome. And as well as this, we've got another key term that we want to mention called ribozymes. What is a ribozyme? It sounds very similar to a ribosome. Well, a ribozyme, if we take a look at this small diagram at the bottom, a ribozyme is RNA within the ribosome which has a catalytic function, meaning that it's involved in specific reactions happening within the ribosome. So an example of this would be in helping form the chain of polypeptides, right, from different amino acids. So in this diagram, this link between amino acids is formed by a ribozyme called peptidyl transferase. That's peptidyl transferase. So that's an example of a specific ribozyme and what it would do within a ribosome. And this specific one, peptidyl transferase, is located within the large subunit of the ribosome. So that's a quick summary of the structure and the function of a ribosome.